The overall goal of the following experiment is to change the permeability of track-edged polymer membranes in UV or white light by modifying the surface with photochromic molecules. This is accomplished by first treating the polymeric membrane with plasma to induce radicals on the surface. The second step is to add a methanolic monomer solution to perform the surface-initiated polymerization on the membrane. Next, the grafted polymer is post-modified with photochromic substances. Ultimately, permeability measurements of the light-responsive membranes are used to show how UV light changes the caffeine permeability through the membrane.
The main advantage of the plasma-induced polymerization is that covalently bound coatings on membranes can easily and homogeneously be created in a reproducible manner. Visual demonstration is critical, as in our custom-made setup the membranes are allowed to polymerize immediately after plasma treatment under inert conditions.
This method helps answer key questions in the fast coating of surfaces and brings covalently bound functionalization to the surface. We first had the idea for this method when we talked to specialists in the field of plasma coating. Beforehand, we had unsuccessful attempts with classical coating methods such as spin coating and dip coating.
Generally, individuals new to this method will struggle because plasma functionalization is a complex technology due to the many parameters that can be adjusted.
Demonstrating the procedure will be Katrin, a student, Karl Kehl,